Hey everyone, here's Miss Larson with a cell membrane discussion description video. In this discussion, you will act as a teacher. Here's what to do. First, open this Google Slides presentation, and I'm going to show you what to do after that. So, open the Google Slides presentation, and what you're going to do here is you're going to go to File, make a copy of the entire presentation. Instead of where it says copy of cell membrane discussion, you're going to put your name. So, like I would put Miss Larson, and you would just put your name. And this is going to make it even easier. You can click share with the same people, and that's going to make it easier for me to be able to see your work when you turn it in. So, then you would click OK. And it's going to make a copy of this Google Forms or Google discussion board. It's on Google Slides for you. So once you get here, you are a biology teacher who just taught students about osmosis. Now, you give them a practice problem to check for their understanding of this new topic. You ask them to complete an osmosis beaker problem, kind of like the one you saw in Edpuzzle. When you see each student's work, you notice that some students are having a hard time. You need to provide your students with meaningful feedback. So tell your students if they are correct or incorrect, and then, if they are incorrect, explain in about 1 to 2 sentences what went wrong and what they should do to fix their problem. When you're done, click share and email me, Miss Larson, a copy of your work. You can even type KLARSON, it'll pop up right there. The other thing you could do if you didn't want to do that would be to click copy link and then paste that into the discussion board. It is completely up to you how you turn this in. You may email it to me, or you can copy the link and put it in the discussion board. Your choice. All right, so reminders about osmosis beaker problems are in the next slide just to help you. So if you go to the next slide, you'll see, okay, osmosis is the diffusion of water. It is a type of passive transport. You have to remember about this thing called tonicity for beaker questions. So when you look at the beaker questions, you always need to look at the solute concentration and not the water concentration. You need to know that hypertonic is where there's a higher concentration. Hypotonic is where there's a low concentration. And isotonic means equal concentration. And lastly, you need to remember that water always moves to where it is hypertonic. And you can see the little problem here. Here there was 10% salt, and there inside the cell was 20% salt. 10 is lower than 20, so that was hypo. Hypo is low. 20 is higher than 10, so that was hyper. Hyper is higher. And water moved into the cell in this case because water always moves to hypertonic, where the solute is higher. So, what you'll do is after reviewing, you will look at this. This is the osmosis problem that you assign to your students for this particular discussion. You want your students to identify each cell as hypertonic, isotonic, or hypotonic. Then identify each solution as hypertonic, isotonic, or hypotonic. Then identify which direction the water will move into or out of the cell. So you're asking that somebody would say, is this cell hyper, iso, or hypo? Is this beaker hyper, iso, or hypo? And will water move into the cell or will it move out of the cell? And you could choose to work this out on your own on some scratch paper and then approach the problems, or you can just look at the problems. This is up to you. So you'll look at Maria's work and you'll determine, was Maria correct? If not, you'll answer these questions. What did the student do wrong and what should they do to fix it? Next, you'll look at Aaron's work and Sheila's work. And that's it. When you're done with that, you will go ahead and share with me or copy the link to put it in the discussion.